In this video, we'll carry on looking at nucleic acids and the various forms of them. In the last video, we had a look at DNA and its structure and some of its packaging. Let's just mention quickly RNA. We will come back and talk about the production of RNA. But just like DNA, it contains a number of features. It contains the sugar, and in this case, instead of a deoxyribose, it is ribosugar, so RNA is ribosugar. The other thing is uracil. We find in RNA, and that replaces the thymine, which we see in DNA. It's a single nucleotide chain, not the double helix. And overall in length, it's much shorter. It's only a very small portion. Once it's copied from the DNA, it only represents a very small portion. And it can have less than, say, about 100 or maybe up to 10,000 or more base pair molecules. And there are three main forms of it. We have messenger RNA, mRNA, ribosomal RNA, rRNA, and transfer RNA, tRNA. Now we're just going to talk a bit about what a gene is. In the past, we really believed that a gene coded for a protein, and in the past it was even said one gene codes for a protein. We now know that's not true because there's way fewer genes than we originally thought in the whole human genome, which is basically a collection of all the genes. And we have millions of proteins in the body, yet we only have about 20,000 genes. So really, what is a gene? Here's a current definition of it. A gene is an information-carrying segment of DNA that codes for the production of a molecule of RNA, and that RNA plays a role in the synthesis of one or more proteins. And the amino acid sequence of a protein is determined by the nucleotide sequence of the DNA. And most of you have seen this in previous biology courses. Humans have 46 chromosomes, and that comes out to 23 pairs of homologous chromosomes. And of course, one set of the 23 chromosomes comes from each parent. The genome is all the DNA in the 23 chromosome set. And to give you an idea, how many nucleotides are we talking? We're talking over 3 billion, about maybe 3.1 billion of them. Just to define the term, genomics is the study of the whole genome and looking how genes and the non-coding DNA interacts with each other and how that plays out in the functioning of the whole organism. Now, definitely Homo sapiens, that's us, have fewer than 100,000 genes. Typically the numbers you see quoted are somewhere between 20 and 30,000. And as we said earlier, a single gene can code for many different proteins. And if you wanted sort of an average ballpark, how many nucleotides contribute to a gene? Well, it's quite variable. Some genes are very small, some are very large. But if you needed a ballpark average, it's about 3,000. But it could be up to 2.4 million bases. Now I like this next bit. And the next bit is that if you look at all humans, we're about 99.99% genetically identical. We're a very young species with a very small founding population. And that means that we're very tight. So almost everyone you walk by is almost a clone of you. But even with us being 99.99% identical, if you're looking at 3.1 billion nucleotide base pairs, then just that small little difference can still represent a few million base pairs differences between everyone. And in general, it's what we call single nucleotide polymorphisms. That's a big word to unpack. It basically means most of the changes aren't that there's huge areas of say, a chromosome that are different between one person and the next. It's usually very one or two letters in certain regions that account for most human variation. Now, the other thing is when we look at chromosomes, some chromosomes have a lot of genes on them and some chromosomes don't have much genes. So it's not like the chromosomes have genes equally spaced between them, that everyone kind of gets 4 or 5, etc., whatever. So there's quite a lot of variation as we go. Now, just a little bit about genomic medicine. This has become much more important in medicine, is understanding a person's genes and how they interact with the individual. This can have a role in terms of diagnosing disease. It could also have a role in the susceptibility you may have towards a disease. Certainly Alzheimer's, there's a genetic component, it's not 100%, your susceptibility to other things, heart disease, etc. And we know over about 1,400 disease-producing mutations, and I'm sure we're going to find many more as we keep exploring the human genome. Now, one thing we're getting better at is by using genetic technology to identify susceptibilities to disease, because we now know that certain alleles, an allele is a variation of a gene, may predispose you to certain things like heart disease, coronary artery disease, certain types of cancers. Now, we're going to start talking about, how do we code for these proteins. Now, the nucleotides on DNA and subsequently on RNA can code for the amino acid sequence of proteins. So really, DNA is largely about holding a code to make proteins. It doesn't make lipids directly, it doesn't make carbohydrates, it's the proteins that we make, such as enzymes or structural proteins in our body, that really go on and allow us to interact, make, and utilize all the other types of organic compounds. Now, when we're looking at proteins, so the millions of types of proteins that we can make are really based on a language where there's only 20 letters. So in terms of amino acids, there's 20 amino acids, and it's just the sequencing of these amino acids differently means that protein either becomes it becomes a protein for you that becomes keratin in your hair, or becomes an enzyme which will do some type of metabolic process. Now, the minimum code to symbolize 20 amino acids is 3 nucleotides per amino acid. Now, that gets us into the concept of the base triplet. I'm just going to hop ahead and I just put a little diagram here. If you think about it, if all we had to deal with are, our A's, our A's, our T's, our C's, and our G's, if one nucleotide was sort of contained the secret message as to what an amino acid would eventually be in the protein, well, we'd be very limited, because at a given position here, what are the options? I could have an A, or I could have a T, or I could have a C, or I could have a G at this particular site. Now, if you think about it, that's only 1, 2, 3, 4 letters. There's no way I could use just one single nucleotide to basically differentially code all these different amino acids. These are amino acids that I kind of put up here. So that wouldn't work, because all I'd be able to do is code for potentially only 4 of them. Not going to work. So how about if we looked at 2 nucleotide sequences? Once again, if you think about it, I could have 4 possibilities here, A, T, C, G, and I could have 4 possibilities here, A, T, C, G. And if you did your math, is that basically if I only looked at 2 base pairs, I could only have a maximum of 16 possibilities. Well, that would still leave potentially 4 amino acids without any code. And we're going to need to reserve some of these codes to say start making a protein and stop making it. So we're going to need stop and start signals built in here. And of course, I'm pretty sure from previous courses you realize that it's actually 3 adjoining base pairs that contain the secret message as to what an amino acid would be. Now, if you do the numbers, this actually comes out to 64 possibilities. So if I look at 3 of these together, I end up with 64 possible combinations, because at this point I could have ATCG, I could have ATCG here, I could have ATCG, and if I worked out all the possible combinations, it comes out 64. Now that's good, because there's 20 amino acids, so that's more than I need, and it gives me some what we're going to call codons extra for start and stop. Now, I'm just backed up and just going to mention this. So there's a base triplet, and that's what we just talked about, the sequence of 3 DNA nucleotides that stand for one amino acid. Now, a codon, and people often use these interchangeably, but they're not quite the same. So when I'm talking a base triplet, I'm talking about the 3 nucleotides on DNA. When I start mentioning a codon is the 3 base sequence on messenger RNA. So there's a slight difference, base triplet versus codon. As I said, it gives 64 possible codons that represent 20 amino acids. Now, out of the 64, 61 code for amino acids, which means some of the codes, we have more than one code possible combination for a given amino acid. So there's a little bit of redundancy built in. And I also have a start codon and a stop codon that are also built in there. I wouldn't ask you to memorize the start codon or the stop codon its sequence, but it's a lot of AUGs, UAG, UGA, and UAA, right? But don't memorize that, as long as you know that there's start codons and stop codons that are also built in the 64 possible codon combinations. Now, protein synthesis. Think about all body cells, except for things like the mature gametes and some immune cells, contain identical genes. So ponder this for a second, that a liver cell has all the same genes as a kidney cell, yet a kidney cell has different enzymes than a liver cell. So cells have to have the ability to select which genes get turned on. So a liver cell will make liver-type enzymes, hopefully, and a kidney cell will make kidney-type enzymes from the genome. Now, just as a reminder, I'll say it again, almost all cells in the body have all the instructions that every cell needs, but cells are very selective as to which genes get turned on or off for their particular needs. So different genes are activated in different cells, and any given cell uses about 1/3 to 2/3 of its genes. So that means there are some of these genes that must remain dormant, and some of them are functional depending on the needs of the cell. Just going to talk very briefly about the flow of information in protein synthesis, and then I think what we'll do is in the next video we'll walk through protein synthesis. There's some very good videos that I put up on our class website that you should definitely watch for protein synthesis, DNA replication, etc. They have animation, which I think makes it fairly clear as to what the process is. It may take a couple watchings of it, but definitely spend your time to get it sort of in the images and the steps in your mind. Now, protein synthesis, really the flow of information is DNA to messenger RNA, which then gets turned into a protein. And there's 2 steps. And don't confuse the steps, even though they have very similar names. Transcription. Transcription is to copy. Just transcribe is to copy. In transcription, DNA codes for messenger RNA. So that's transcription. Occurs within the nucleus, and the messenger RNA sequence is complementary to the gene. So it's based on the instructions in the gene. The next one is translation. And when I say translation, I'm talking about going from the messenger RNA code to proteins. And in this case, the messenger RNA migrates from the nucleus to the cytoplasm, where it'll code for the amino acids. And we'll talk more about that in detail. So we're going to come back in the next video and briefly talk about transcription and translation. But once again, I highly encourage you to spend time looking at the videos that are posted on our website.